A paper entitled Human-Chimpanzee Differences in a Frizzled 8 Enhancer Alter Cell Cycle Dynamics in the Developing Neocortex by Lomax Boyd and colleagues in the Ray and Silver labs appeared in Current Biology. This study is a landmark towards understanding how DNA changes could have led to evolution of the human brain and our cognitive abilities. In this BioEssays piece, Lucia Franchini and I critically review their study and discuss future directions that may help us link specific DNA sequences to traits that distinguish humans from other species. Hello, my name is Katherine Pollard, and I'm a senior investigator at the Gladstone Institutes and professor at University of California in San Francisco. I will be discussing a review article that Lucia Franchini and I wrote for BioEssays entitled, Can a Few Non-Coding Mutations Make a Human Brain? The human and chimpanzee ancestor lived about 6 million years ago, and since that time, both species have acquired traits that distinguish us from one another. For example, we've learned cooking practices and gained enzymes that enable us to eat a diverse diet, including dairy products and high-starch foods such as grains and tubers. Our bones changed to enable upright walking, and we lost anatomical features associated with hanging in trees. Interestingly, some of the most devastating diseases facing our species, including atherosclerosis, neurodegenerative diseases, AIDS, and certain cancers, occur uniquely in humans or at a much higher rate in humans compared to non-human primates. I want to understand these unique disease susceptibilities by studying what makes humans different from chimpanzees. Some of our most intriguing differences as a species relate to our cognitive abilities, including long-term planning, abstract thinking, and our elaborate spoken language. Changes in the human brain and both its structure and its organization likely facilitated evolution of these uniquely human traits. So what is unique about the human brain? The most striking feature is that an exceptionally high proportion is devoted to the cortex, the center of higher thinking. For example, more than 84% of our brain mass and more than 20% of our neurons are located in the cortex, which is a higher percentage than any other species. There are also unique structural features of the human brain, such as connections between neurons that are different. And we are seeing these now through new non-invasive brain imaging techniques. As we continue to identify features that are unique about humans, an important question arises. How are these differences encoded in our DNA? It's important to understand the genetic basis for human traits because this knowledge will shed light on the evolutionary forces that shaped our species and that differentiate closely related species in general. It will also help us to understand how these differences may have affected human-specific diseases and pinpoint the genes and pathways that are involved. For instance, HIV researchers would like to understand how chimpanzees and other primates can tolerate immunodeficiency viruses, whereas humans cannot. This information will help us to figure out a cure for AIDS. Unfortunately, understanding the genetic basis for a human trait such as this is hard. A decade ago, I made a discovery that laid the groundwork for the study of Boyd and colleagues. We had just sequenced the chimpanzee genome for the first time, and I developed a computational method to screen the 300 billion plus base pairs of the genome to identify sequences that are conserved across other species but different in humans. These human accelerated regions, or HARs for short, turned out to be almost entirely outside of gene sequences of the genome, in what we called at the time junk DNA. So could a few non-coding mutations really have led to emergence of the human brain and our cognitive abilities? I thought maybe so. So we and others hypothesized at the time that these sequences, these HARs, were regulatory elements and that they might be controlling the expression of genes that are important in the developing embryo. We didn't know very much about gene regulation at that time, but since then we've learned more. Specific sequences have been tested for their regulatory activity, and we've designed genome-wide screens to predict where regulatory elements lie in our DNA sequence. And excitingly, it has emerged that HARs are indeed largely developmental enhancers, in line with our original prediction. Boyd and colleagues picked up on this work and designed experiments to specifically test if human accelerated enhancers could have altered the human brain during evolution. First, they designed DNA sequences that carried either the human or the chimpanzee version of the accelerated enhancer, and they showed that the human version drives a different expression pattern in the developing neocortex compared to the chimp version. Next, they showed that this sequence physically interacts with the promoter of the frizzled 8 gene, a developmental transcription factor. This suggests that the sequence regulates the expression of frizzled 8. Finally, they put the mouse frizzled 8 under control of either the human or the chimpanzee version of the accelerated enhancer, and they showed that the human version leads to a faster cell cycle in neural progenitor cells and a larger cortex in the mouse after birth. These findings are very exciting, but there are caveats. First, because the frizzled 8 gene and the human accelerated enhancer are located close to the centromere, it was challenging for the investigators to change the actual mouse sequence to either the human or the chimpanzee sequence. Consequently, they instead inserted an extra copy of that DNA into the mouse genome. Because we do not know from their data exactly how many copies inserted or where in the mouse genome they landed, it's difficult to know for sure if the differences that were observed in the developing mouse brains were indeed due to the human-specific DNA changes. Second, they only studied human and DNA sequences and no other primates. Without the other primates in the study, it's difficult to know for sure if the differences that were observed were due to the human DNA sequence differences rather than DNA changes that were specific to chimpanzees. Finally, both the human and the chimpanzee sequences were studied outside of their native context in a mouse. I'm excited that many of these caveats will soon be addressed with emerging technologies. A technique developed by my colleague at Gladstone, Shinya Yamanaka, has enabled us to take a skin biopsy from a human or chimpanzee and make induced pluripotent stem cells and then differentiate these into neuronal cells that are human or chimpanzee. This provides a platform to finally be able to test human-specific DNA changes in a primate. Another technique, called genome editing, is making it easier for us to modify the DNA of these cells and of model organisms such as mice. Finally, high-throughput genomic techniques, such as the massively parallel reporter assay, are enabling us to test if HARs or other DNA sequences alter molecular, cellular, or organismal phenotypes. Lucia Franchini and I are optimistic. With these techniques in hand, we will soon be able to know if a few non-coding mutations altered the human genome during evolution and led to uniquely human brain features and traits.